Hello, good afternoon. It's Doc Kyle, and this afternoon we're gonna talk about the nucleus. So remember, the nucleus is the center, so kumbaga, or the brains of your cell. Um, if the cell is the basic building block of life, meaning it needs to have a control center, and the control center of the cell is the nucleus. It dictates what proteins are to be done, are to be created. What are proteins? Proteins are your biomolecules that make up for the structure of the cell, the secretions, their products, no, and then their overall structure and physiology. They are quite dictated by your. They are dictated by your proteins, and these proteins, the creation of these proteins, are dictated by the blueprints that are found in the DNA found in the nucleus. So this is a cutie pie nucleus, no? So with great power comes great responsibility. So the nucleus needs to take care of the DNA. So but if it needs to take care of the DNA, therefore it needs to have a membrane. So this is a nucleus, no? So what sorry. You can see here the nucleus has a um it has a wall. It's a double wall. What makes this nuclear envelope special is it protects the DNA from the rest of the cytoplasm, meaning the DNA is well protected. So the DNA can be found in your chromatin. Actually, your chromatin is just strands of DNA wrapped around your histones, which we will talk about later. And then these are your nuclear pores by which your mRNA can transfer, can go inside, or your hormones, your steroid hormones, or your kanang nuclear hormones can go inside and attach to the nucleus, no. So this is your rough endoplastic reticulum. So the rough endoplastic reticulum is rough because it has these ribosomes. The ribosomes are in charge for the translation, no, translation of your mRNA to your proteins, no. So the central dogma is translation, transcription, translation, and protein synthesis, no. That's your central dogma. So what are the functions of the nucleus? Cellular regulation, houses genetic material, like I said earlier, the DNA, and which directs cellular activities, and then the production of ribosomal units in the nucleolus. So the ribosomes are created in the nucleus. So they are in charge of protein synthesis. So the nuclear envelope. So the the inside of the cell is separated by the nucleoplasm by the nuclear envelope. So it's in charge. It's a very selective membrane, kumbaga, meaning it doesn't let anybody in or anything in. Doesn't let your cytoplasmic proteins go in. It doesn't let your toxins from the cell go in. So kato, no? So this is our central dogma over here. You can see this. So we'll wait. The step 2 is translation. So the first step in your central dogma is the first step is your transcription. The transcription, always remember that your DNA is a double helix. So it is a pair of nucleic acids, no? So the steps, and then what happens is you open that double helix, and transcribe a part, meaning transcribe a part and convert it into an RNA. And then your RNA from the nucleus goes outside to the cytoplasm and meets with your ribosomes to create your protein. No? So that's the translation. You translate your RNA into your respective amino acids, forming a peptide. A peptide is combinations of your amino acids. No? So the other name for your protein is your polypeptide. All right. So the nuclear envelope is penetrated by nuclear pore complexes. So they're in charge of letting the RNA move in and out. So the nuclear envelope is, of course, supported by your meshwork. As you can see here, this is the meshwork, that red Kaning red din na area. So these are your nuclear pores. They are in charge of the transport of or the movement of mRNA through the nuclear membrane, no? In and out. Next. So this is a presentation of, think of the green ones, the green lines are your DNA. No, they are your DNA, and the red balls are your histones. So they wrap around your histones, and that is your chromatin na, your chromatin. So this is the larger. So what is a chromatin? Chromatin is what colors your nucleus, no? Chromatin, if simply put, that your DNA is like thread, and then the histones are like spools, your chromatin is the thread plus spool, no? Muna siya. And then if you combine it together, that's and you compact it, that's what makes your nucleus colored, because the usual stain that we use, stain pang color sa ato ang mga slides in histology and histopathology, is hematoxylin and eosin. Always remember that hematoxylin is an alkaline stain, meaning its pH is alkaline. And in biology, if there is opposites attract, so an alkaline stain will bind to an acidic compound. And from the name itself, DNA, the A is acid. So the hematoxylin will bind to the acid, no? So making the nucleus color blue. And then the second stain that we use, hematoxylin, no? And then the second stain is our eosin. Eosin is an acidic stain which binds to the alkaline compounds. Usually your proteins are alkaline. That's why your proteins are commonly seen in your cytoplasm. So in your cells, you will see blue ang nucleus and your cytoplasm red, because most proteins are alkaline, and ang eosin will combine to the alkaline compounds, which are your proteins. So remember, the thing that makes your nucleus blue is the increased amount of your DNA, no? I know, not the increased, the DNA itself. So kato. So this is your DNA. Like I said earlier, this is your double helix, and then this is your histones, the red one, and they pack together. That is now a chromatin fiber, no? And then there is a protein scaffold here. Protein scaffold. And this is your. So there are two types of chromatin, no? We have the heterochromatin and the euchromatin. I said earlier that in your central dogma, in order to have protein synthesis, no, there must be transcription. But it's very hard to transcribe your mRNA, no, from your DNA if they are very compressed, like this one in your heterochromatin. It's really condensed, no? Daso kayo siya. But so it needs to, the heterochromatin needs to unravel gamay, so that the DNA will be exposed, so that there will be transcription. So the unraveled chromatin is now your euchromatin. It's a light staining because it's unraveled. So the active chromatin is your euchromatin, and your inactive is your heterochromatin. So you gets, kay nag-unravel naman siya, nag-transcribe na siya, active siya, it's actively transcribing. So it's an active type of chromatin, your euchromatin. Na? So kato. So if you're gonna have a mitosis, the cell will have undergo mitosis. So of course it doesn't, if mag-mitosis na siya, mag-cell division na siya, dili na, it's not expected to undergo further protein synthesis. We'll talk about the cell cycle later, but it's not expected to produce more protein synthesis, no? So, but it needs to compact itself. That's why everything will condense, and most in mitosis, tanan na lang na mo na chromatin will become heterochromatin. It will be very condensed, and it will form your chromosomes. This is a chromosome for mitosis, no? So that's what happens. Becomes a chromosome in mitosis. But if after na mitosis, na divide na siya into two, after telophase, it will begin to unravel, no? So because it needs to build more after manggod mitosis, wala pa manggoy unod ng usually ng cytoplasm. So na nagoy unod, but it needs to create more structures. So eventually it will unravel to produce more proteins for the cellular structure. No? Okay. So chromatin is the combination of DNA and its associated proteins, like I said earlier. So that is active in transcription is your euchromatin and ito, like I said earlier, this is a presentation. So the extra X chromosome in the female mammals, because in females it's XX man, diba? And the males is XY. So it's quite redundant na it's not needed na in the functions of the body. So it will become just a condensed body, cellular body. That is what you call your bar body. So that's you can see. We can see that in your neutrophils sa mga babae and your oral mucosa. So makabalo ka na babae ang patient if makakita kag bar body, no? So we have types of heterochromatin, the constitutive heterochromatin. So it's repetitive. So it's the actually it's just more of filler jud siya, or required jud siya na naa jud siya sa it constitutes the chromatin jud, no? While so it's the ends of your chromosomes, no, and your centromeres. It's like part of a structure jud. While your facultative heterochromatin contains other regions of DNA where transcription is variably inactivated, no. So your facultative heterochromatin, there is a chance that it might be transcribed, while your constitutive, it's more or less it's poor transcription jud siya, no, wala na jud siya chance na i-transcribe, no. So okay, so kana imahang that's a clue. So your facultative heterochromatin has the faculty to be transcribed. No, it has the option, while your constitutive it just constitutes something, the structure of the chromatin, no. So this is your nucleoli. So always remember that your nucleoli is always there. No? So your nucleoli is the site for your RNA production, no? So it has yeah, siya nuclei nato. The nucleoli over here. So usually in a normal staining cell, you don't see the nucleoli. But if the cell is very it's very active, physiologically active, increased iyang cellular iyang protein synthesis, iyang activity, then the most of the chromatin will be converted into euchromatin. And if it's euchromatin, then you will see your nucleoli there na. But if the cell is quite dormant, you won't see the nucleoli because it's mixed together with the heterochromatin. So the nucleolus is a very basophilic, electron-dense area of chromatin. No? So it's a compaction of your chromatin. So that's where RNA transcription and ribosomal units are assembled. No? So kato. So the cell cycle, no? The cell cycle is a sequence that events control the growth and division of the cell. So you have your G1 phase, the longest part, no? This is the most variable cycle of your cell cycle. So this one occurs immediately after mitosis, no? After nimo divide, this is G1 dayon. So in G1, ang imong ginabuhat, you're trying to recuperate after division, because always remember when a cell divides, gitungaan ba ita ng cytoplasmic proteins and structures ana sa duha ka cell, no? So it needs time to fill up those cells na ay mga cytoplasmic structures na wala, mga organelles na wala. And then we have your synthesis phase, the S phase, no? The S phase, kano siya, is the DNA synthesis phase. The S means synthesis, meaning you're duplicating your DNA in preparation for your mitosis, no? So after G1, na replenish na tanan imong cellular imong cytoplasmic organelles, the DNA now is trying to duplicate itself in preparation for the next mitosis, no? So kato synthesis phase, no? And then the G2 is the preparation to the G2 is the preparation for your cellular cytoplasmic preparation for your mitosis. It prepares the proteins needed for for mitosis, katong mga katong microtubules needed, 'di ba? Na kay mga opposite na polar body na mag-pull apart sa imuhang cell. So, okay, remember, G1 is recuperation after mitosis, S phase is your DNA synthesis, and your G2 phase is your cytoplasmic preparation for your mitosis. And then, M for mitosis. Gamay lang kayo ng mitosis compared sa tanan, no? So, G1 is the most dependent, no? It's the most nutritionally dependent, di ba? Kaya mag-recuperate pa man ang cells, no? So, if you're malnourished, you don't have ma-prolong jud imong G1 because it takes time to produce proteins needed for your cytoplasmic na recuperation, no? Yeah. So, this is your mitosis, no? So, you can see here, so interface after nimo unsa ayun, S phase DNA record. So this is your G1. Your G1, it's very long compared to the other na mga mga phases, no? But there is a branch, is quiescence or differentiation. So your quiescence meaning wala na siya buhaton, mag-chill na lang siya. Di na siya mag-mitosis, kanang mag-rest lang jud siya, wala na siya buhaton. Or differentiation. This differentiation means na mag-mature siya, magka-gain siya og more functions, dili niya priority ang mag-divide, no? So example, in your skin there is a kanang germinal layer, kumbaga, na siya ang gikan tanan mga cells na supply. So what happens there, na yung mga cells there that are always undergoing mitosis to replenish the skin, because remember that your skin will eventually be sloughed off, no? Malugod man na sila eventually. So there's time na pero na po yung ibang population na mature jud mahimo yung keratinized na skin cells, no? So kato. And then this is your S phase, your DNA replication, and then your G2. And then ano yung mitosis nimo? So mitosis is also kanang siya mitosis. Very short, your prophase, your PMAT, no? Prophase, metaphase, anaphase. Ako ipa-remember lang ninyo, metaphase is your equatorial plate, no? O lang yun na siya. In your prophase, your nucleus starts to lyse, no? Na nakikaryo kanang karyolysis ana kay maggawas naman imong mga chromatin. Magtunga na siya, magline tunga imong mga chromatin sa imong metaphase unya magbulag na sila together in your anaphase, no? The chromatins will now split together. Always remember na nag-divide na sila into two na, di ba? Kay doble-doble naman na imuhang DNA kay nag-S phase naman ka. So in your telophase, there is already a cleavage formation, no? Siya. So kato. So the nuclear envelope disassembles in your prophase and your microtubular spindle forms. Metaphase, katong chromosomes are aligned. Itong anaphase, nagbulag na jud ang imuhang mga chromatin. And then your telophase, katong there is now cytokinesis, no? There is cleavage into two cellular daughter cells. So kato akong ingon ganiha na yung mga cells na always will divide, no? That's their goal. Kana lang jud ang purpose, mag-divide. Sige lang sila mitosis, no? And then na yung mga ubang cell na mag-differentiate, no? So we have this when you look at over here, this is your stem cell, and then they will divide into two, di ba? Kaya mag-mitosis sila. But one cell matures, differentiates, this is the differentiation, and it becomes a columnar ciliated cell lining sa imuhang intestine. While the other one is still a stem cell. It just divides. So that's the purpose of your stem cells, no? To replenish the population. Okay, always remember that types of cell, especially your epithelial cells, they just the purpose of your epithelial cells is just to protect, and they eventually get sloughed off, no? Matanggal lang jud na sila, malugod lang jud na sila. Kaya especially sa imong intestine, always remember na sahay ba di ka nakausap tarong sa apple, no? Kana mo ani mo na usap ng apple, so gahi pa kaya na siya, so magasgas na imong tinai. So mas slough off jud ang imong epithelial cells dito. Same with your skin, di ba? Manglugod ka, you will exfoliate jud. Alright, next, your stem cells and renewal. This is another kanang representation of your stem cells. So wala lagi na siya. In tissues with stable cell populations, most connective tissue, smooth muscle tissue, and differentiated cells undergo slow and episodic division to maintain integrity. So, your stable cell populations are your most connective tissues. Wait lang, butangan ako na siya og picture. Alright, no? So, I have a picture na magic. No? So, this is your cell cycle. No? So, we have a labile cell. Meaning, pag labile ka nang, di siya mapuyo ba? No? So, sige lang siya, divide, no? That's your stem cells, no? But, na yung mga cell na after division, especially pagka nang even fetal pa ka, nasa fetal stage pa ka, na after niya divide, it will differentiate immediately. And then, that is your permanent cell. No? Example is your neurons and your muscle, meaning they don't undergo mitosis anymore, no? So, they are permanent cells, no? So, that's what you call differentiation. Differentiate na siya. So, once you damage your neurons, it's bye-bye na, no? Tumuuna, mga tao na magka-stroke, no? Di na maulian. That's why, no? Paraplegic na dyan sila. Di na sila kalihok. Di na sila kalibang. Di na sila kalibang. Di na kalakaw. Kina-stroke naman. Di naman mahil ang neurons. There's another type of cell. Your after... So, kanino? This is your stable cell. Immediately after your mitosis, no? They go differentiate and they become stable cells, meaning they still have the option to undergo kanang mitosis, but they're stable, chill lang sila. They will only undergo mitosis if there is kanang damage to them, there is damage, or there is a need to do more, no? So your liver is a very resilient na organ, no? You can still live even though 20% na lang imong liver na buhat, no? Ay nabilin na lata tong 80%, 20% na lang nabuhi. It will you will survive, and furthermore mag-regenerate pa jud na imong liver. So it's a very kanang resilient organ. So an example of your stable cells is your hepatocytes. All right, next is, so if there is mitosis for your somatic cells, in your sex cells, no, namely your sperm cell and your oocyte, no, your oocyte, no, we have meiosis. What happens in meiosis, mangod? So remember, in meiosis 1, it's divided into meiosis 1 and meiosis 2. So in meiosis 1, you undergo the same steps as your mitosis. You have prophase 1, no? Mag-break down ang nuclear envelope, and then they have metaphase 2, no? Mag-align sa perlahi ang sa meiosis. Duha ilahang double file sila versus your mitosis na single file lang. And then you have your anaphase 1, ano? Your anaphase 1. So your anaphase 1 or your telophase 1 ay mag-divide na sila. What happens is the cell, since duha man sila na file, dili man sila kanang single lang, whole na chromosome ang ilahang madala individual cells. Kay in mitosis, half chromosome lang man na ilahang madala in mitosis. But in meiosis, it's just a they take the entire chromosome. Kaya nag-double line man sila sa metaphase nimo instead of one sa imong mitosis. So, in their anaphase and telophase, they bring to each individual cell is a whole chromosome. And then mag-metaphase 2 na po na sila. Mag-metaphase 2, pag metaphase 2 na, mag-single file na sila sa metaphase 2 nimo, magtunga na sila. So overall, your endpoint in your meiosis 2, you have haploid, meaning you have half your chromosomes. So the total chromosomes in a somatic na cell is 46, but in your after your meiosis, your sex cells, your sperm cell and your oocyte is only 23. That's because 23 imong ginadala sperm if you're a man, mag-meet na sila sa oocyte sa imuhang partner, sa wife nimo, mahitabuan na mag-combine sila to form another 46, which is unique 46 na set of chromosomes, that's the offspring na, that's gonna be the embryo, goods, so this mo lang to siya. Alright, so half jud ang sa unsa. Look over here, 46, tanaw to sa meiosis. You have starting at, we're gonna look over here, we're gonna start at 46, then we have 23, so your endpoint is a haploid, because the usual common is 46, also known as your diploid, so apoptosis, it's simply put na this is the suicide of the cell, apoptosis simply puts is example, if a cell can't go on, there is extreme damage to it that it cannot repair itself, and then it will undergo apoptosis. And then the gene for apoptosis is seen in your mitochondria. Fun fact that your mitochondria was separate na unsa siya sa una, in the prehistoric times, it was a separate microorganism, so anyway, ana lang na siya. If di na niya kaya, magpakamatay siya. Example, pag grabe na kayo, mag-apoptosis ang imuhang, example, na damage ka, na sunburn, na igo kayo imuhang mga cells, mag-apoptosis na lang sila mo, mag-peel off na sila. That's why mag-shedding ka, mag-exfoliate ka kung ma-sunburn ka, excessive kayo ang damage. So versus your necrosis is a pathologic death, apoptosis is a physiologic, meaning pag physiologic cell death, normal siya, kanang expected siya na mahitabo, versus your necrosis is your pathologic, meaning pathologic disease na siya, dili siya normal na cause of death, meaning kung magka-necrosis ka, usually na yung mga factors na ma-necrosis ka, gidunggab ka, kana necrosis na siya, kanang nagka-infection ka, namatay ang mga cell, gipatay sa kagawang cell, that's necrosis. And then, hanap po. So, so apoptosis is physiologic cell death. Sorry. And then, so this is a practice quiz. So, what characteristics does the nucleus make it basophilic, So, lagan siya acid, meaning yung pH kay blank, So, what cell cycle is highly dependent on nutrition, Katong, di ba? Katong ingon ganiha, katong directly after mitosis, Child was born with fused digit. What physiologic process did not occur during fetal development? Like I said earlier, ang imong kamot manggod kay fused manggod na. So, na yung mga cells diha nabuhi pa. There are times na kailangan yun na siya, physiologic na siya, napatyon yun na to cell. So, what is the physiologic cell death, And malignant cells with high rates of proliferation and division are expected to have. So, kato. So, what is this increased mitotic figures, So, of course, kasi man siya proliferate, so increased din yung mga mitotic figures. Maulang mga mitotic figures, those are the figures that we see during microscopy. We can see the metaphase, Pag makita natin sila sa line, that's the mitotic figures, or nagtunga na sila, nag-divide na sila in your anaphase and your telophase, That's your mitotic figures. So, what is this synopsis, So, what is this synopsis, Synopsis is when there is gonna be shifting chromosomal shuffling rather, chromosomal shuffling. And this usually occurs in your prophase 1, no? So, sa prophase 1 na siya mahitabo um, mag chromosomal shuffling. So, what's the point of chromosomal shuffling? If always remember that a genetically diverse organism, no, be it a kagaw or a human, is more resistant to your to infections, no, to death. Kay same with your COVID good na sige siya mutate. Adlaw-adlaw man na every minute nagamutate man ang COVID, no. But sometimes it mutates for kanang forced for better, no? In the sense of COVID, but in our case, it's for the worse, no? Meaning, same with your Delta variant na mas grabe siya maka-divide, mas grabe siya ka-infectious. Same with us, humans. So, if we shuffle our chromosomes, we might get a gene that is very kanang healthy for us, no? So, example, in Africans, no, sa mga atong mga igsoon dito sa Africa, they have a gene that makes their blood resistant to malaria. That's only endemic to them. So, through the years, nakadevelop sila inato na gene through shuffling. And, ma-preserve tong gene na to kay ang mga nabuhi lang tungod kay wala namatay sa malaria because they carry that gene, no? And then, apart from that, there is some kind of mechanism na if that gene is always used, especially nagabuhat siya proteins because of that gene, no? Because of that chromosome DNA na unsa na kanang pattern, sige siya buhat na protein based ato na pattern, ma-preserve na siya. So even if mag-shuffling, dili na siya malabot sa shuffling na, no. So ma-preserve siya or ma-enhance pa siya, no. So further down the line, so mao na siya. Kung ikaw namatay ka tungod kay wala kay inato na gene, meaning weak imong DNA, it's like the survival of the fittest, no. So mao na siya. That's the point of synopsis. That's why it's a genetic lottery, kumbaga. So, that's all. So, if you have questions, just feel free to PM me. Dali lang mo ko istorya. And, ito. See you. Bye-bye. God bless. Good night.